Thanks, everybody, for sticking to the end. And I would like to also thank CPTR for the invitation to be here. I have been supporting Critical Path Institute with regulatory support for several years now, and in particular CPTR for the last 3. I'm here representing the Health Authorities Submissions Group of CPTR. And our remit is to provide regulatory expertise and support to the various working groups across CPTR as we engage with regulators and decide on the regulatory strategy and pathways. So I'm happy to see some of you here in person that I've heard you on phone calls several times. I'm also happy that in that capacity, I get to work with Bob Clay, who's here today. We also have 2 other members of the HAS group who couldn't be here today, Karen Rombarts from Janssen, although I've met some of her colleagues here, as well as Karen Ragland from Otsuka. And I will take this time to make an advertisement that we are also looking to increase this group because we are going to have a lot of exciting regulatory opportunities coming up, and that's what I'm going to be talking to you about today, regulatory pathways that we will be pursuing at CPTR. It's really hard to make a title of a regulatory talk sound exciting. So I'm going to do my best to hope that that comes through during this talk, because we really do have a lot of interesting interactions coming up. And I think that's also evidenced by the participation that we have throughout this conference by our FDA colleagues. So we've had several of you here today, and then we will have some throughout. And so this is a very interactive and exciting time as we pursue some of these regulatory qualifications. Let me make sure. What I'm going to do today is quickly give you a high overview of the qualification process, and I'm going to be able to build upon some of the examples you've already heard about. So I hope it will come to life for you rather than just being a list of what some of these regulatory pathways are. And then I'm going to use 2 examples that you've heard about, one from the past, the hollow fiber system, to make some of these, give you some more details about some of these examples and make some points about those, and a new and upcoming one that we just heard about from Larry, the LAM biomarker qualification efforts. And again, as we've heard many times today, one of the overarching remits of CPATH in general and CPTR in particular is to accelerate the timelines and increase the efficiencies of drug development. And we do that through a variety of methods, and we've talked a lot today about the importance of I'd put it in different order. I'd say data sharing and then data standardization. You need to share it first, and then we've got to get it standardized. And I think we've all heard how important that is, and there's such a strong expertise of that is supported by the CPTR and CPATH in general. I'm going to focus on accelerating things through biomarker development qualification. There is also an equivalent pathway for clinical outcome assessment measures and qualification. And I think just listening to the fascinating summary that we got today from the REFLECT-TB also can show you how important having some of that same type of standardization and perhaps even qualification efforts eventually on clinical outcome assessments is really important. So we won't talk about a lot of that here, but it is another very similar pathway to what we're looking at in biomarker qualification right now. And then obviously the modeling and simulation, we heard a lot about that from our last group of panelists. So both the FDA and the EMA have an official process for this type of qualification. In the FDA, we call it Drug Development Tool, or DDT qualification. And in EMA, you can't see it, but it's called Qualification of Novel Methodologies. They came out about the same time. These started in very early 2010, 2011. Their procedures are similar. They're not exactly the same. They're 2 different organizations, but they really have tried hard to also harmonize them. And you even have an opportunity to actually go through both of the processes of qualification in a parallel manner if you choose to do so. Even if you don't choose to go parallel with FDA and EMA, the work you do for one is highly transferable to the work you do for the other, even if you do it separately. What I would also like to talk about with respect to there's the similarity is some of the differences between the two agencies with respect to qualification. And in particular, I think probably the biggest one is that there are logistical differences. So with the FDA, you have no fee involved, but you also have no dedicated timeline involved with respect to when the interactions occur and when you get that feedback. With the EMA, you do have a fee that you need to pay to get the qualification advice. They do give you the possibility of a SME, a small and medium enterprise fee, which is a much lower fee to be able to do that. And then you do get a very specific timeline for getting the feedback that you want. Very similar, if any of you have gone through the scientific advice, it's the same timeline on which they work. And we've seen this, I think, this definition, but I'm going to take a little bit of time on this slide just to talk about the qualification. It really is a formal regulatory pathway, and it provides acceptance of biomarkers for their use in drug development. But I want to make the point that while this is formal, it really is very interactive with regulators. And some of you, again, in the drug development world know what it's like to go to request formal meetings, type A, type B, type C, the type of data that you have to present when you go towards those meetings. So there's a lot of data, there's a lot of rigor involved, but it is also a very interactive process with the FDA on a different type of interaction level than there are with true IND, NDA drug development. And the other thing I think it's very important to talk about, and we've, again, heard examples of this, so you're making my job easy up here, is that the qualification is always tied with a very stated, specific context of use. And it's within that context of use that then the biomarker is qualified, and that when it's used in that specific context of use, it can be applied throughout drug development. Not just for a specific drug, and not just for a specific IND or NDA pathway. And I think we saw two really good examples of just talking about the importance of a context of use. When Dr. Garner was talking about the PBPK context of use, we've actually laid out about 3 of those and how we picked those out. I think those are very good examples. And then Larry with the LAM, how we've really worked hard to come up with a very specific, clear context of use on which to base our qualification process. That's critical. The other thing that I think is that is important to focus on here is that we have. I think if you really look at the first bullet point here, is that once it's qualified, you get information that's pertainable for and acceptable to use in biomarker development, and it becomes publicly available. So in the case of the FDA, the final outcome for a qualified biomarker is that you get a draft guidance attached to that. And so there is then public availability of that information. The biomarker becomes publicly available. That doesn't mean that it has to be free. You can still have one that's used and maybe has some proprietary information attached to it, but it still becomes, the information becomes publicly available, and that's a very important aspect of being able to share this across the drug development world. For the EMA, they also, built into their qualification process is a public comment period. So if you get to qualification, then there's a period where the public is able to comment on that opinion before, and the FDA, and the EMA takes those comments into consideration before they issue the qualification opinion. I've got to grab water. I talk fast. And then what I would like to talk about a little bit, I should have maybe labeled it even differently on the slide, is really what biomarker qualification is not. And the first one is that it is not just biomarker discovery or one time of a clinical validation of your biomarker in your lab, but it's a formal acceptance by the health authority. So as such, it has to be built upon a lot of data. And if any of you have heard some of the quotes that Janet Woodcock herself makes about this, is that the work that has to go into the, there's a lot of very interesting and exciting things that happen at the discovery level, but the work that has to go into then really validating it, replicating it across sites, in different people's hands, maybe across different subpopulations, depending upon how you're going to use it, that is the work that has to happen over and over and over. And it's not particularly sexy. It's not funded by NIH. So it really takes groups like people in this room to come together and be able to basically donate their time to do this on top of your other day jobs to get the work that is done. And that's why I think it is so important. The consortium model is it takes a village to do this, because you really do need people contributing the data and then being able to go through some of this replication that you need, so that you can have the credibility and present that to the FDA, because that's what they need in order to be able to approve this. So much as some of the modeling and simulator team we're talking about up here about the importance of credibility, it's the same thing that you need for the models. It's the same thing you need for qualification, to have that additional type of work that goes forward. And then the last thing that's on the slide just makes the point that when you're going through qualification, you're not qualifying a particular tool to be used as an approved device, for example, as an in vitro diagnostic. You're qualifying a biomarker to be used, again, across platforms within that context of use. The other thing I didn't put on here that it's not is it is not a requirement. So once that a biomarker becomes qualified, it doesn't mean you have to use it. It just means it is there and available as one of the gigantic toolbox, or maybe not so gigantic, but we're trying to make bigger toolbox for TB drug development, or any drug development. And again, this just goes into a little bit about some of the things I think we've already talked pretty clearly about the objectives for qualification. But one of the things I want to talk about is in the final point is that this provides a framework for scientific collaboration, and I should actually say in their regulatory collaboration to facilitate drug development tool work. And I think this is very important, even if you don't have qualification as your final endpoint. And I think we had some really good examples of this today, particularly with Eric's discussing the sterilizing mouse model. Even if you're not stating up front that your goal is qualification, if you use some of this process to approach the work, your work is then regulatory ready, or at least it's more regulatory ready than it might be in a typical academic process when you are looking at some of this scientific work, and that is really invaluable. It's never wasted work. It really helps you with your own program, and then if you decide you want to use it in some type of regulatory application, sticking to these processes will be very valuable in that type of work. So I think it's a really important framework above and beyond whether you do or don't decide to qualify a particular biomarker. And this is just, I'm showing this, I think we've talked about these three levels of information that you need to do. Start out with a very defined context of use. Gather your level of evidence, and then get your biomarker qualified. That makes it sound really simple. It's not, but that's what the steps you need to take. And when you look at the biomarker qualification guidance, particularly in the FDA, they refer to this as the three stages of the process. So you've got an initiation stage, and that's where you will be submitting something that's called a letter of intent. Fairly small document that you provide to the FDA that says, this is what we're going to do, this is why, this is the context of use we're going after, these are the questions that we have. That's the initiation phase. During this level of evidence phase, you're really, and they call that the consultation and advice stage, and that is a process where you are pulling together information, putting it into a briefing package, and putting it in front of the regulators. And that can be a couple steps in there. That's why these are circles, because sometimes you circle back around. You give them an initial briefing package, they review it, and then they come back and tell you what else they need to see it. And then eventually, you submit your full qualification package, and then that's the final stage, which they call the review stage. And so I really put this up here to emphasize the reiterative process, that this is. Also to share with you some advice that we've received repeatedly from the FDA as we go through this process, which is to start narrow and work wide. And again, I think that is something that you bite off what you can, what you've got the best data for. So, for example, we've heard again today, started off with the hollow fiber system because that's what we had a lot of data for. With the example with the PBPK that we heard about, we're going to start off with 1, 2, and 3 context of use, and then you can widen those. It's a very similar process to what we do in drug development with IND, NDA. You get your initial approval, and then you tack on additional indications, different populations, and you can continue to do that with the biomarker qualification process. But starting small, in this relatively new process in the scheme of things compared to drug development process, will serve us well. And also the starting with the end in mind, which is something that usually is very helpful no matter what you're doing. And again, in the drug development world, I equate that to working with your TPP, your target product profile, where you start at the 2 pages, say this is where we want to get to, and then you fill in the blanks to get to that. That's what we're trying to do here. And I don't have a slide for this, but one of the what I do want to stop here with respect to a question that we get asked so much: why qualify? Why go to this? Why go to the work that it takes, the collaboration that it takes to do this? And to me, the answer is because you can go down the one drug, the individual company working directly with the regulators to get a biomarker used in a clinical trial. That data will eventually, information will eventually come out, and people will learn about it. But when you do it upfront, collaboratively, trying to qualify a biomarker for a broader use with colleagues, and then having it become publicly available, it is something that really helps move the field forward. And you have to have some level of, in my mind, a vision of the greater good, not just for you or your company, because it really does help move fields forward. And this happens in fields like TB, where there's maybe limited resources and limited people in the field, but we've also seen it happen successfully in very competitive fields, like Alzheimer's disease. We've had some consortia, and again, it's because you can do more together. And so I do think there are really both scientific as well as altruistic or greater good applications for this type of qualification. Now I'm just going to go this will be very quickly because we've actually talked a lot about these, but I'm going to talk about a couple examples, one from the past and one from the future. The past one is what we've done with the hollow fiber system. Again, this is just we've seen this several times now, just showing what we do before we get started, gathering information, setting our context of use, working through that process. The other thing that happens as we talked about in the hollow fiber system was bringing together data and bringing it together and then getting it into a standardized system that we can use. A lot of times the endpoint for that is the application of CDIS standards, but not every area has CDIS standards, but it is giving some type of standardization and structure to the database so that you can use it for your future analyses. And then the other point I want to make from the regulatory side of this, the hollow fiber system, you've seen this slide again many times. And I don't know if you can see the dates, but this is a good example of where we did not go parallel at all. CPTR started going through with the FDA, starting with the example of an LOI, letter of intent. Eventually, there was a decision that let's look at this in a different context with the VXDX, which is voluntary exploratory data submissions. Got input on what might be the best way to take those data that were being collected and use them. In the end, those data, as you've heard, the outcome of that type of regulatory interactions was submission of that work as comments into a guidance. So in the end, it almost happened the same way it might happen with a qualification, where you get a guidance on it. Here, at least, this was also mentioned in a specific guidance that reflected that's being developed now for the TB. And then after that was done, we were able to take that fully developed package and then work it through the EMA. And so, and then with that, in that case, we really did go through a full qualification process. And it probably doesn't jump out at you if you don't do this every day, but what this represents from February of 2014 until January of 2015 is a first time through qualification, because this package had been so well developed by the CPTR team and the groups that were there supporting it that it got through on a qualification the very first time. So this was a very successful, maybe not straightforward and not parallel pathway, but it really got us to two very, very important regulatory endorsements, one through the qualification and one not, but still has been very useful for the field. And the next example that I want to talk about, whoops, sorry, I think I went backwards, is the one that Larry was just talking to us about LAM and that regulatory interaction, which is a biomarker qualification. I'm not going to go into this. It's I should have done it earlier, but it doesn't matter. And what we are doing here is looking, as Larry described, at a pharmacodynamic biomarker that we're trying to qualify. It's the first, at least at CPATH, it's the first time we're going straight to a pharmacodynamic. Most of the biomarkers that we've done, and actually the ones that have been qualified, are either prognostic or safety biomarkers. So this is a jump for us. And again, Larry went through most of this, so I'm not gonna reread that. But there are a couple things that I do want to say about the CPI meeting on this slide. Larry reviewed it and also provided us with his testimonial. That it really, regulatory interactions really can be fun. I mean, I'm biased, I'm a regulatory professional, and I love regulatory, but these are really very interactive and very helpful meetings. The other thing is there are, even though these are relatively new, they started in 2013, there are regulatory guidelines for this. There is a very interactive website. I also should have said that about the actual DDT documents. Both EMA and FDA have just excellent websites that really give you a lot of information about how to qualify, give you good examples, so that's a very good resource. And the other thing that I want to talk about with the CPIMs that we didn't get into is that the resources involved with them are relatively minimal. You need to have basically a five-page letter requesting the meeting, and so that is very different than what you have to do with some regulatory programs in the IND-NDA pathway. And you take it in front of the regulators. When your meeting is accepted, you give them a slide set. So there's not a large briefing package that goes in with this, not a ton of data. Your data are basically being presented there in a slide set, and so that also, I think, really does help with the interactive activeness of it. The next thing, we've talked about these examples. I will tell you right now that we are at, since 2013, we are at, the FDA is at 41 CPIMs they've had. 25 of those happened in 2016. So these are really ramping up, and they're really a good start for the qualification process. But we can anticipate that the waiting is going to be a little bit longer as we go forward, because they are just getting a lot of them. And I think this is, again, some of the details I've already actually mentioned in other, as I've talked through other slides, and Larry showed this slide. Again, the FDA does come back with you after the CPIM. They have 60 days to give you a summary. And it's not as detailed as you might get within a IND meeting, but it really, you get what you need basically at the meeting itself, which was very, very useful for us. And finally, I just want to end with some conclusions about the biomarker qualification in general. They do provide better insights into how to progress through drug development. The qualification process lets you use these across multiple development programs, and your regulatory acceptance of that is assured. You don't have to keep going back to the regulators asking for that. Once it's qualified, you know you can use it. This is something that is difficult for a single organization to do, so it really does help, even though the collaboration takes time and energy, it really does help to move both the data and the field forward. And as we do so, we're also working to help regulators and ourselves understand what evidentiary considerations are necessary. What do we need to show as we do this? It's still a work in progress as we go through this biomarker qualification process. And finally, that this allows a lot of data sharing with our key stakeholders. It allows novel biomarkers to be used in a variety of stages, including late-stage clinical trials, to help assess some of the outcomes and hopefully enhance some of the outcomes. And finally, during this whole process, we do keep a very collaborative relationship with health authorities that we establish and maintain throughout the process. And it's, as I said, it's reiterative, so hopefully it's a long-term interaction that we'll continue to be having. I think I'll stop there, and Larry, you want to come up and